So, using cancer genome sequencing data, we have identified 25 patients that have HER2 mutations in their breast cancer. You heard from Dr. Picard about the role of Herceptin and the importance of HER2 in breast cancer treatment. When we look at these mutations, see if I have the mouse here, you can see that they cluster in two areas of the HER2 protein. A large cluster here in the tyrosine kinase domain, which is the active part, the enzymatically active part of HER2, and a smaller cluster in the outside half of HER2. Patients with these mutations will be missed by current HER2 testing. Gene sequencing is required to identify these mutations. And as we went forward with our research, our hypothesis was that patients with HER2 mutations will benefit from anti-HER2 drugs. This is a summary of our research results, and you have this in your handout. So the HER2 mutations predominantly occur in patients who are HER2 negative by conventional testing. These mutations are found only in the breast cancer. They are not inherited, and I want to emphasize that fact. 13 mutations were tested in the lab using direct enzyme activity measurements, tissue culture experiments, and tumor growth in mice. The majority of these HER2 mutations are activating events that cause cancerous growth of the cells in the lab, meaning that they stimulate the function of HER2 or turn on HER2's activity in an excessive or inappropriate manner. Thus, we feel they are highly likely to drive the growth of these human breast cancers in which they occur. My fourth point, one of the HER2 mutations, the L755S mutation, which was found in 6 of the 25 cases, nearly a quarter of the patients that we looked at, was drug resistant. It was resistant to Lapatinib, but the growth of cells containing this mutation could be blocked with another anti-HER2 drug, Neratinib. And I'll show you data on that in the next slide. So what you're seeing here is an experiment where we test the concentration of drug that's needed to inhibit cancer cell growth. And we're comparing Neratinib and Lapatinib. The numbers that are shown here, the lower the number, the better. The lower the number, the more potent the drug. So what you can see for Neratinib is it requires very small concentrations of Neratinib, about 2 to 15 nanomolar, to inhibit the growth of cancer cells containing these HER2 mutations. In contrast, much higher doses of Lapatinib are required, ranging from 400 to 1,000 nanomolar. And let's see if I can get the mouse. Here we go. The L755S mutation, as I mentioned, is resistant to Lapatinib. It continues to grow even at 10,000 nanomolar of drug. So because of this greater potency of Neratinib and its ability to inhibit all of the HER2 mutations that we've tested, we've moved Neratinib forward into a phase 2 clinical trial. This is a multi-institutional trial for patients with metastatic breast cancer who have HER2 mutations. The eligibility criteria, stage 4 breast cancer, and have to be HER2 negative by conventional testing. Their tumor DNA will be screened for HER2 mutations. If the mutation is present, they will be treated with Neratinib, and response to the drug will be measured by serial CT scan. There are 4 institutions participating in this trial. This trial is open at Washington University School of Medicine in St. Louis, and will be opening soon at our other sites, Dana-Farber Cancer Institute at Harvard, Memorial Sloan Kettering Cancer Center in New York, and University of North Carolina in Chapel Hill. Perhaps my most important slide for the purpose of this press conference is this one. So the significance. These results show how cancer genome sequencing can be directly applied to guide individual patient treatment. HER2 mutations are a target for breast cancer treatment, and gene sequencing is needed to identify these patients. A multi-institutional phase 2 clinical trial is now open to screen patients for HER2 mutations, and if they have the mutation, treat them with Neratinib. And if this trial is successful, we estimate that 4,000 women per year in the U.S. could benefit. Worldwide, that number could be 5 times as large. There are many people who contributed to this work. I'd like to thank my colleagues at Washington University School of Medicine, Charles Peru at the University of North Carolina, colleagues at the NCI. I particularly want to thank the patients who took part in the genome sequencing studies, and the full scientific report is published this morning online in Cancer Discovery. Thank you.